Hello everyone, I am Raphael Mourad, Associate Professor at the University of Toulouse, and I will talk about the last tutorial that I've done for large language model for genomics. So this is the training here, and you'll find it on my GitHub. Uh, so the last tutorial is about optimizing a DNA sequence. Can be also protein, mRNA sequence, but here I will do DNA sequence optimization for instance, transcription factor binding, or it can be also used for promoter optimization. Just have to change the title here. So let's just click on the Google Colab, and I will comment the lab class. So here we are. So let's check whether we have a GPU or not. Me, I don't have any more GPU because I used a lot of GPU for doing all of these tutorials. But it's fine. We don't need, I mean, it's not compulsory to have a GPU. Also, it can speed up computations, of course. Let's do it. So it says the device is the CPU. We are downloading the GitHub because in the GitHub we have some fine-tuned models. I will talk more about that. That we will use these fine-tuned models. You will see how do we optimize a DNA sequence. We'll check the system version, I mean the Python version. We have to install a dataset Python module, so we'll do it. I mean, do we really need that? I think we don't, but anyway, I have to move that from the script. Anyway, so let's import modules. So transformers, torch, numpy, and of course, what we mean by optimizing, it means that we take an initial sequence that we take. It can be a random sequence as initial value, initial seed, and then we'll optimize the DNA sequence toward uh, using an objective function, so using a criteria, so that our DNA sequence will fit to this criteria, and we'll try to maximize, for instance, the criteria or minimize the criteria. So it can be the binding of a protein, or it can be the fact that we have a promoter. We can do promoter optimization. Okay, so for instance, here we'll use a differential evolution algorithm from SciPy Optimize. Okay, so the differential evolution, we'll talk about that more. It's it's stochastic in nature. It's looking at large areas of space candidate space. So actually, it's running a lot of, it's testing on many DNA sequences by modifying the initial sequence a little bit. It's like mutating the DNA sequence, by the way. So let's load that. Let's say we want to optimize a promoter. Okay, so we want to make a DNA sequence that is a promoter. So you can take a DNA sequence that has a low promoter activity, and then you can try to optimize it to have a higher promoter activity. So it has many applications in molecular biology. If you have a gene and a promoter, okay, the corresponding promoter is a gene, and you find that this gene is not expressed enough, you might want to modify this promoter to optimize it to make it a stronger promoter so that the gene is more expressed. So the optimization task is promoters. In this case, we'll load a large language model, Mistral DNA, with 1 million parameter that was pre-trained on the human genome and that was then fine-tuned on a binary classification task uh, with a set of sequences that are promoters, okay, that are known to have promoter activities, and a set of sequences that are known not to have a promoter activity. So we'll use this model, okay? And this model is able to predict, given a sequence, it is able to predict whether it is a promoter or not. So we'll load that. So for that, we load it with auto models for sequence classification. So it means we're loading the model architecture together with weights. that were trained to predict well whether a sequence is a promoter or not. Okay, so let's load the tokenizer. Here is the objective function that we are defining. So what is the objective function? So what you have to keep in mind is you want to maximize the score of a sequence to be a promoter. Okay, so when you use what I did when I trained my model, I set up the non-promoter sequences as 0, labeled as 0, and the promoter sequences as 1. And then my model, when it sees the DNS sequence, if it predicts a value close to 1, it means it's a promoter. And if it predicts a value close to 0, it's not a promoter. So, but when you optimize with this with digital evolution, you have to minimize the function. Okay, so it's set up like that. The premise is set up like that. So what you have to do is just minus. Your model predicts something, and you want to take minus this value. If you're predicting it's a promoter, so it's a value of 0.99, you take the negative value so that instead of maximizing, you're minimizing. Okay, it's just because the algorithm needs to minimize instead of maximizing. So we have this findings objective function. What you have to do also is here, this is not, so when you optimize, usually when you optimize in a continuous space. So you have variables that can take continuous values, okay? Here, keep in mind, this is a DNA sequence, so it's a discrete optimization. So your letters can be only for a given position. A letter can be only A, T, C, G. So this is a discrete optimization, not quantitative. Okay? So discrete qualitative optimization. So it makes it more complicated. So what we do is we define bounds, and the values are between 0 to 3. Okay, so example of bounds for each character. Okay, so I have a function that converts a DNS sequence to values that are 0, 1, 2, or 3. Okay, for each position. We'll see. So it's num2sec. So yeah, so here this function is converting, sorry, it's converting numbers, so 0, 1, 2, 3 to ATCG. Okay, and this one, sectuname, is the opposite. It's converting a sequence with ATGC to numbers 0, 1, 2, 3 for each position. So here's the coding here. You can see it. So we're gonna define this function. Also, I made a function to generate a random DNA sequence of length k. So it is here. Here is a function to predict the score. So meaning whether it's a promoter or not. If it's not a promoter, the score will be close to 0. If it's a promoter, the score will be close to 1. So what you have to do is you have to convert the sequence into numbers. Then it will be tokenized. Sorry. So you have to convert the numbers into sequences, and the sequence will be tokenized. And then from the tokens, you will predict the score. Because keep in mind, so the model takes a DNA sequence, okay? It converts it to tokens with the tokenizer, and then it predicts. But when you will be optimizing, you will not optimize directly the letters of the DNA sequence. You optimize the corresponding numbers. And I will show you. You will optimize the corresponding numbers. So it means that during optimization, so you will have numbers, so you have to convert the numbers into the sequence, and then from the sequence you will give it to the deep learning model, to the large language model, by tokenizing the sequence and then by predicting. So that's what we're doing that. So for instance, just to check that the model is working well. So I have a negative sequence, meaning that I have a sequence that I know it is not a promoter. So it's a negative sec. I convert it to the numerical representation of the sequence. and then I predict the score, okay? And I do the same with the positive sequence for which I know that it is a promoter. I convert it to a numerical vector, and then I predict. So let's have a look. So here, my sequence is T, G, G, C, C, A, blah, 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 okay? So a T is a 3, a G is a 2, a G is a 2, C is 1, etc. So I just converted this sequence here to this sequence here, okay? And then I give that to predict score function that will convert it back. Predict score will convert back to a DNS sequence and then tokenize it and then predict. Why do that? Because inside the the the, the differential evolution optimization algorithm, it takes numbers. It doesn't take text. It doesn't take letters. So, so that's why I'm just testing my function, if it working or not. So what I can see that for the negative sequence, the score is 9%. It's not really a probability, but you can see it as a probability. So it's just a probability of 0.09, which means it's not a promoter. And for this sequence, which is my positive sequence, this is encoding in numbers, and this is a prediction. So 95% chance of being a promoter. I say just be careful. This is not really a probability. It's kind of pseudo probability, but let's say that it's a probability for the ease of interpretation. So I'm just testing my objective function here. So here is just to test that the function that I defined were good, and I give numerical vector here, and this is the corresponding. And here num2sec is just if you give a set of numbers, this is a translation into a DNA sequence, okay? So let's start. Let's start. So I'm gonna start with a random. So I'm gonna generate a random DNA sequence of length k equal to 15. And then I will take this random DNA sequence and I will mutate it basically. I will change letters. For each letter, I will change them. It will be kind of evolution because it will mutate every letters, okay? I will do that for many sequences at different steps because I will mutate a sequence once and then a second time and a third time if the score improves, the score of being a promoter gets improved. So let's do it. So here is my random DNA sequence when it is translated into numbers. So just to say a 2 is a G, 1 is a C, 0 is a A, 3 is a T. So basically it's G, C, T, A, G, G, A, etc. Okay, so this is the sequence. I didn't make it that long because it takes a lot of time if you make a long sequence, just to say. Uh, This is the corresponding score of being a promoter. So it's kind of low. Just keep in mind, if you run it on your computer, you will find a different number. Okay, you might find a value of 0.8, for instance, because randomly you got a sequence that has a high score of being a promoter. This is really possible. And if I rerun it, I will get look, I got 60% here. If I run it, I get 13%. Let's keep with 13%, just to say we'll have different values. So I did that. Then these are the boundaries. As I said, my values are from 0 to 3 possible values, okay? With 0 is A and 3 is T. And 15 is the length of the sequence, okay? So let's start with that. And we'll use differential evolution. So they say it finds a global minimum of a multivariate function. The differential evolution is stochastic in nature. So it will randomly go into the space of DNA sequences by mutating my initial sequence. It will mutate once and then twice and a third time. Uh, just to say, this is what is called a black box optimization. We have a neural network. From a neural network, we can compute the gradient of of the difference between our DNA sequence, the current DNA sequence, and the objective we want to have. We can we can we can compute the gradient depending on the input. So we could use a gradient to optimize, and it could be faster. Okay, here we're using a very, in a sense, a very black box approach that works with, in the cases we don't have a neural network, it works with many cases, many scenarios for which we have a model. We can predict a value given an input. Okay, so you can change the strategy from that because we have values by default. Yeah, I use many values by default. Population size 15, so we keep a population of 15 sequences. We have a recombination, so it's an evolutionary algorithm with mutation. So it means it will mutate at each position, the letter. It's great because this algorithm can be applied to many situations when it's not a DNA sequence. Okay, but here it's a real DNA sequence. So actually, the evolutionary concept that really makes sense here. That I'm just saying because this algorithm could work with just optimizing the number of cars, the number of cars in the parking, which has nothing to do with the DNA sequence, but you'll still have mutation and recombination parameters inside the algorithm. Here, the great thing with the DNA sequence is the mutation really is a mutation. So it's like mutating a letter, and recombination is you have different solution, and then you'll merge to some extent, you will combine the different solution together. And it makes sense from the DNA point of view because it means you'll take two sequences and you will just recombine them. So you take a piece of one DNA sequence, another piece, and you will just concatenate them. So it's pretty nice to use this function for DNA optimization because it's really very intuitive. Although this algorithm generalized to many situations that have nothing to do with DNA. So let's do it. I used I'm using just default parameters, just to say, just a maximum of iteration of 20 so that I don't wait for hours. Let's do it. So just to say, I'm not a very big expert on optimization. So I'm using, I will say I'm using a function. I know many concepts, but I'm not really the expert in this topic. So if you have many questions, I might not be able to answer all of them. But I can show you here how to use it. So it's pretty nice. We're starting from a DNA sequence that is not a parameter, and we will modify it with many mutations and recombination. And we'll see that at the end, we have a DNA sequence that has a higher probability of being a promoter. So let's wait. It's testing like here, we don't see it, but yeah, it was testing many, many solutions. So here we have, look. So because it's negative, just remember that was minus a probability of being a promoter. Actually, the probability of the best sequence we got of being a promoter is 96%. So we went from 13% to 96%. So it's pretty nice. It was pretty easy to optimize the sequence. I just wanted to say this algorithm might not work with large sequence. We might need to use more sophisticated approaches in gradient descent or other approaches. We have a population of DNA sequences. We're actually, I'm not extracting the population of solutions we got, but we have a population of solutions, just to say. Uh, all right. So you can, for instance, inside the result object, you could dig inside. I'm not gonna dig inside. And then what I know is the best solution is here, result.x. This is the best solution. So keep in mind, it's made of numbers, 0, 1, 2, 3. And then I'm gonna convert. So I'm gonna decode these numbers into a DNA sequence. Let's do it here. So this is a DNA sequence. Here it is for promoter. And here I just show you what I got for promoter. So before I got something that looks a little bit like that. That's a bit different. And then let's compute the score. Okay, 96%. So just wanted to say, we took a DNA sequence. So what you could do actually, if you're working in molecular biology or in genomics, you have a promoter or you have a sequence or you have a promoter, let's say, that is not very efficient, and you want to convert it to a promoter that is more efficient in expressing the gene. You could use that to increase to mutate a little bit the promoter to increase the chance of expressing a gene. So this can be used. I mean, promoter optimization is used in plasmid to optimize the promoter inside the plasmid for gene expression. It's very used. Okay, so this is how you could do by using a fine-tuning LLM. Okay, so let's go to optimizing transcription factor binding site. So it is a DNA sequence where a transcription factor binds. Usually in a promoter, there is one or more transcription factors that bind to the DNA sequence. So here it's based on ChIP-seq data. So ChIP-seq data, it's based on ChIP-seq experiment. ChIP-seq are chromatin immunoprecipitation experiments where using an antibody, it targets a DNA sequence that binds a protein, which is a transcript factor. And then I have a set of sequences that bind the transcript factor, the TF, and sequences that don't bind the TF, that don't bind the transcript factor. So to do that, it's just simple in the tutorial. I'm just gonna modify one parameter instead of promoter. I will change TF binding. Let's do it. So now that's the name of TN binding. So it's still a large language model, Mistral DNA, but that was fine-tuned for TF, okay, for TF binding. Let's load the model. So the architecture of the model is the same, but the parameters are different. Uh, The functions are still okay. Here, this is a DNA sequence that is known not to bind the DNA. This is a negative sequence. This is a DNA sequence that is known to bind the DNA. So for the sequence that is known not to bind the DNA, you have a probability of 3% of binding the DNA. And for a sequence that is known to bind DNA, you have a probability of 85% of binding DNA, okay? We can just do that also. Let's generate a random DNA sequence here. Here, just by random, drawing a random DNA sequence, we already have a probability of 80% of binding. So now I have 24%. I prefer 24%. I prefer to have a DNA sequence that has a low probability of binding a transcription factor so that I can improve. Let's do differential evolution. Let's go. So you can see it's quite quick, by the way. We are running on a CPU, not a GPU, and already achieving good results. So you can make, if you're working on your own computer with your own GPU, I'm sure you can increase the length of the DNA sequence. They optimize because usually a DNA sequence, yeah, it's quite small. So let's do it, and we'll see. Basically, it's the same thing as we did before, but with another fine-tuned large language model. And my aim was also to provide you with a model for mRNA optimization, but the model I have for some reason is not working, it's bugging. So I will work on that later on to see if I can provide you a model for mRNA optimization. For instance, to optimize the stability of the mRNA or the protein expression of the mRNA. But for now, I cannot show you that. So here we are. So we got a sequence with 92 probability of binding, a TF. Let's get the numerical vector and translate it to a DNA sequence. Here it is. So for instance, you can see it has a lot of G's. You have stretches of G's here. Likely it is important for the TF to bind. And let's compute the score, and you have, as I said, 92%. Okay, so here you have the beginning of an optimization script where you can optimize in sequence. Of course, you can do the same with protein sequences. As I said, with RNA sequences, you can do that with many things. You have to change the model, and of course, you have to change to adapt a little bit the function that I've done. All right, so I hope that you find it clear, and don't hesitate to write some comments. And if you have questions, to write some questions. Thank you very much.